So as you can see on the screen, this is a very non object that you can see here. It is a neuron cell. I am faculty of mathematics, but I can tell about little bit biology and how mathematics is useful in this biological area. Context with the mathematics. So once we talk about, I will take 10 minutes to introduce how it's work. So as we know, our nervous system that works throughout the body, but it is maintained by the brain itself. The brain execute the orders, give the orders, and receive the response also. So as you can see in our nervous system, the human brain and the spinal cord, these are the main major part of the nervous system, and it's termed as a central nervous system. So basically, our nervous system divided in two parts, CNS and PNS, central nervous system and the peripheral nervous system. Central nervous system consists of the brain and the spinal cord, and rest the part that is the peripheral nervous system and a number of nerve cells that can be seen in our human body. For example, if you talk about the heart, these nerve cells exist inside the heart, that is cardiac myocyte. In same way, the hepatocyte is there. If we talk about the pancreas, so SNR cell is there. So all these cells are responsible for the movement of the information that we use the word neurotransmitter. Information, that is the term as a neurotransmitter. But how it works inside the cell, that is again important part of a study. So as we go inside the brain, billions of neurons you can see inside the brain. Neurons are the basic building block of the nervous system. It works very smartly, but it not work alone. A number of supporting cells exist in our human brain. For example, if we talk about glia cell, astrocyte is a part of glia cell, oligodendrocyte, number of swan cells are there. They are supporting the neuron. So first we talk about the neuron. As you can see on the screen, the left part, it is a dendrite is given. Dendrite is rece- responsible for the receiving the neurotransmitter. It means neuron receive the neurotransmitter information from the dendrite, and then it execute inside the nucleus. That we talk about the soma, or sometimes we say the cytosol as well. So our main work is on the soma. Here the information executed, and then it goes towards the this axon terminal via axon, and again it release toward the next neurons. So the neurotransmitter that moves toward the next billion of neurons are there. Neurons are connected with the number of neurons. Actually, what happens in the soma so that the neurotransmitter moves from left to right? Just come to inside the neuron cell. What happens here? Inside neurons, number of subpart that you can say, or subdomain you can say, exist here. As you can see here in the figure, the terminals are releasing the neurotransmitter, and number of receptors of other neurons are there, that are these are ready to accept receive the neurotransmitter here, and the gap between these two neurons that term as a synapse. So synapse plays very important role for the movement of the neurotransmitter or information. So what happens inside the cell? Inside the cell, number of parts are there. You can see the nucleus is there, endoplasmic reticulum is there, well-known mitochondria is also there. All take place very important role in the movement of the neurotransmitter. I use the word movement of the neurotransmitter. So what happens inside here you can see. Okay, which parameters are very important for the movement of neurotransmitter? Because uh, in the medical science, clinically, you understand. Suppose a patient means what? The person who is feeling pain, suppose headache or any pain, what they do usually, they generally take the medicine for the pain relief. So what happens inside the brain, inside the body? Actually, once the person take the medicine, the neurotransmitter which is responsible for the pain, which is carrying the information pain, that blocked by that chemical. So that what happens? The neurotransmitter not able to move from one neuron to other neuron or one part of the body to the other part of the body. In that case, the patient do not feel pain. Once the patient not feeling the pain, our immune system just work there, and after some time, the effect of medicine is not there, and the person again do not feel the pain if the pain is such type like headache. But what happens in the pain when teeth about pain again comes? So again the patient take the medicine to release relief from the pain. So that happens in that case. It means what? Some chemicals are responsible for the movement of the neurotransmitter. So as we can see on the screen, the Ca2 plus is here, calcium ions are there. So this is the grace of God. Free calcium ions are responsible for the movement of neurotransmitter. As we know, the 99% of free calcium ions exist inside the bone. Only 1% free calcium ions are responsible for our whole nervous system. Definitely, this is miracle. Only 1% free calcium ions is responsible. Now the question comes, how it's work inside the neuron cell? So that if some physical process is there, if something is changing there, mathematics takes place, and definitely mathematical model that design the process. What is mathematical model? Mathematical model that represent the physical process, the same physical process that we are talking here in the biological or biophysical processes. So come to the free calcium ion. This is the parameter that we have to focus how it's work. Then we talk about some mathematics regarding there. So as you can see, the free calcium ion that coming inside the neuron cell, but it comes via different way. Different way it comes. Here, the voltage gated channels are also there. The sodium calcium exchanges are also there. But in short, what we did first here, that free calcium ions are coming inside the cell. So as it comes inside the cell, what happens? The concentration level of free calcium ion rises. It rises at such level so that these neurotransmitters moves toward the synapse. This is not enough to understand the movement of the neurotransmitter. We can say a lot of calcium ions will come inside, and definitely frequently it will come. The frequency of movement of neurotransmitter, definitely it will go high. Information will move very fastly. It is not like that. If free calcium ion moves inside the cell and the concentration rises higher and higher, it becomes toxic for the neuron cell. Again, at the same time, it required to balance the free calcium ion for the movement of the neurotransmitter and the whole function. So our important parameter that is required to measure, that is free calcium ion. At the moment of ions, two type of signaling happens. First is electrical signaling. Electrical signaling means what? It is a potential difference between inside and outside the cell. And due to the potential difference, channels opens and the neurotransmitter moves inside the moves from one cell to another cell. But if it not works, then the calcium signaling take place. So we are working on the chemical signaling here. So what happens here, if in the second figure you can see, if free calcium ions are coming inside the cell, number of proteins exist there. Number of proteins means what? Number of chemicals that we can understand. That chemicals react with the free calcium ions and makes the calcium bound buffer. This reaction is a two-dimensional, two-way reaction that we say. In that way, it manages the free calcium ion. The role of that chemicals, that proteins is what? Buffer. So in fact, that's why it is also known as buffer. Is it sufficient to make the neurotransmitter move from one side to one cell to another cell? Again, no. Number of inside cells are there, endoplasmic reticulum, mitochondria are there. They are also taking place. Very important role in signaling process. Okay, so if it goes high and high, some part of cell is going to be damaged. That we say the tangle and plaques. Tangle and plaques are generated so that if it goes higher, the symptom of Alzheimer's disease starts. So the movement of the free calcium ion also responsible for the some neuronal disorder that we have studied also there. So the first process, what happens here? Free calcium ion entering inside the cell, proteins are reacting with the free calcium ions and makes the calcium bond. So here the first physical phenomena that we understand that the reaction term is going on. So as you know, the person who is familiar with the chemistry or chemical engineering, if there is a chemical reaction, differential equation take place, and that represent the chemical reaction also. So that we use there some physical law that is required. Okay. Some other, as I told you, some other cells are there. My PhD work was on astrocyte. Neuron also some persons are working. So there's interaction between astrocyte and neuron is a part of the study. Many authors have studied interaction between astrocyte and neurons. So this is another physical process. So inside the cell, again, number of, you can see the channels are there, number of source or sinks are there. Source and sink physically that we understand, the flux inside and outside that goes. So this process takes place here. How it work? If we go for the solution, I will come again for the diffusion process. Number of analytical numerical techniques can be applied for the solution of some diffusion equations. Suppose we talk about the two-dimensional diffusion equation. This is the two-dimensional diffusion equation there. So if we talk about the numerical solution, that the system of equation will take place here, the matrix algebra is applicable. Suppose our region is rectangular region. So in that case, what happens? Here you can directly go for analytical solution because it is a very regular shape, no issue to solve. But if your problem is little bit different, for rectangular finite difference is easily applicable. But our part is to introduce the irregular shape. As we see, the neuron is very irregular, astrocyte is very irregular. We can't compare with the regular shapes. So in that case, what happens? The finite element method is applicable. So this is the beauty of finite element method. I can't ignore finite difference method, because if we talk about the time dependent problem, finite difference method is only the one method numerically that can be applicable for the time unsteady state problems. Other than that, that Laplace transform can be applicable. So these are the some geometry where we can see the irregular shapes are we can generate. So what is happening in the finite element method? You can discretize the whole domain in the small number of pieces. It may be the rectangular, it may be the triangular, anyway. And after solving the problem element wise, again you compile all the elements and make the final solution. This is the beauty of finite element method. So in mathematical neuroscience that we talk about in our area, you're required to go for a study of some biological or biophysical process, apply some mathematics, mathematical model that we require, and some computer simulation that we require. In which way we can formulate the model that is ordinary with the help of ordinary differential equation, partial differential equation, as well as fractional differential equation. Apart from apart from that fractional differential equation, we have started the fuzzy differential equation also. I make it clear these are only the different concepts for the same process, right? Ordinary partial differential equation that we term as a number of independent variables, and fractional if we talk about the derivative, how it's go from first order to second order, zero to first order, that we term as a fractional differential equation. If there is a linguistic uncertainty, then we talk about the fuzzy differential equations. So analytical numerical method can be applicable, integral transform that you can use for the analytic method if the domain is regular. If it is irregular, we prefer finite element method. Finite volume method also can be applicable, and some software that we have used and, applicable there, MATLAB, COMSOL multiphysics that I will show the example also. Okay, so if the chemical reaction take place, A plus B is equals to C with some association and de-association rate constant. So what will happen? Physics is applicable, physical law required to make the this chemical reaction to the mathematical equation. So law of mass action take place. So what it states here, the rate of chemical reaction is directly proportional to the product of the activities or concentration of the reactants. What it's been, if we are talking about change in A with respect to time t, that will equals to minus k1. That is the A plus B is making the C. It is a negative direction. It is reducing the value of A. So, so that it is minus k1 AB plus K2C, because it is bidirectional reaction. So from C again it generates. So it is a plus K2C. Same way, if we talk about del C by del T, it will be reverse order. The right hand side you can see the sign is only changing. Rest is same. We can expand the reaction, the chain of reaction. A plus B is equals to C plus D is equals to E plus F. Something that you can talk about. So that can be the chain will be also designed with the help of law of mass. Some literature. First, I will show here. Then I will talk about the some problems there. So mathematically, the in the neuroscience, the first paper that we can see, it was published in 1993 in Journal of Physiology. That is calcium buffering properties of calbidin D28K and others in rat sensory neurons. So calbidin D28 is the one kind of buffer. It is a protein that react with the free calcium ions. So all about they are talking about experimentally also. They have examined experimentally. In 1996, in 1996, G.D. Smith, he had designed the mathematical model for the calcium buffering. So in he has published two papers in on the calcium buffering process. So the first is analytic steady state problem of the buffering approximation near an open calcium channels. In one paper, he has studied the access buffer approximation, and others he has studied about the rapid buffer approximation. So it was the assumption of the author that the protein is moving or not, protein is fixed. So mathematically also that can be designed. Those who are willing to work in this area, so mathematical physiology, you can uh, go for the first book, the first author. Okay, here the point source that happens. Science, more than 300 article he has published and neuronal calcium. In this paper, he has studied with a by studying this some mathematical process that can mathematical models can be in India. The first that MNIT Bhopal. SG Tiwari, Sivan G Tiwari has published first paper on finite considered. The beauty of these studies, you can ask what happens if we study the math working in US with this study after Wisconsin US. So that very important part. If you go for mathematics and biology together, also working in same university, Medical College of Wisconsin. She is working as a postdoc. So definitely, it is very exciting to read maths and biology together. In another paper, in 2000 studied this paper. Our first paper, genuine paper that we can say, distribution in astrocyte in presence of X. Those who are interested, just go visit this paper about the finite element modeling. After getting this, the first paper that we have published on the calcium dynamics with aligned with the fractional derivative. It is very peer-reviewed journal. Around four impact factor is there, 4 point something. So it is published that time. With the help of fractionally, we have studied the role of free calcium ion on the neurons. Again, in 2018, we have published in International Journal of Biomathematics. Those who are interested in study of fractional differential equation and its application in biology, go to this paper. I definitely recommend this paper because everything we have written in this paper, the reviewer have uh, recommended everything to write. Do not hide any term, any topic, so that we have written there. The first paper that you can definitely study. So again, was key that work on the this calcium signaling with the help of sodium calcium exchanger. The Sivan Tiwari again, he has published some paper. Some important review that I have shown here. So that can be definitely those who are interested in working in this area, the, they can study these papers first. So ordinary OD, PD, fractional we have studied. Fuzzy differential equation also we have used for the same study. Finite element method, finite volume method we have used. Analytically also we have solved. Now we have started on bioinformatics. Again we are extending this to the neuroinformatics. These works are very important for the study of for the beginners as well. These books we recommend for the biomathematics people. Devanshi did the medical college we have started site in this small world. We have the process that is the calcium buffering process. As the calcium enter into this cell, number of proteins are there. Number of protein means that is the chemical substance that react with the free calcium ion and makes the calcium bond buffer. This is the first part. We have not high concentration is there, and inside low concentration is there, and due to that difference that makes the huge difference, the process how it comes in this is your chemical reaction as told earlier. A plus B is equals to C, so it is here Ca2 plus Bj. Bidirectional reaction is there, CaBj. That buffers are there, that's why we have written the Bj. Number of reactions takes place. So once you apply the Fickian diffusion law and the law of mass action, del C A2 plus by del T, change in free calcium ion with respect to time, that equals to diffusion coefficient and the two-dimensional diffusion plus this summation Rj, that is the reaction term. Equation 3 and change in calcium bond buffer that is defined in for think about before going for the solution of the final model. Here we have considered the immobile buffer. Immobile buffer means what? There is no change in that case del b by del bj by del t is 0, and at the same time this is also going to 0. Mathematically if we come we talk about we have considered. So by considering this, what we get here del c by del t, Ca2 plus x square plus dcay del square c upon del y square minus f of x y c. It is internal process. No doubt only the calcium buffering we have considered, but in this part other source and sink also can be considered. Why it is in a different way that can be considered, so that we have initially we have written here. Same equation that can be written here. So this is our the final model to represent the calcium diffusion and calcium buffering. So it is a what? It is a reaction diffusion equation that we termed as. Earlier also it will be the reaction advection diffusion equation will take place. But here I am presenting here the reaction diffusion equation. The boundary condition that is considered here, the so Ca2 plus infinity means what? As it moves towards the out out from the source, far away from the source, then it will be what? Steady state condition, that is in terms of the in terms of domain now. It means what? We are considering the domain is regular. Either it is rectangular or it is square. Circle or your elliptical. So in that case, what happens? We can Laplace transform by separation of variable. Many method can be applicable, and definitely no question for the domain only. So suppose we have such type of domain. We have tried to cover the element technique. Initially, when we discretize the domain of the regular shape, we have used some method like Ridge method we have applied for because once you discretize it, it is very difficult to get the values manually. So in that case, what happens here? The blue line that so can sum into via this way only. Here in the second figure, we have considered the multiple channel. Maybe there is multiple channels are there. Also, you can consider the solo flex and the multiple. Now the next part is what how we can get the solution. First, we discretize the whole domain. As you can see, the number of pieces that we can see here. So initial mesh node is 278 and the 382 triangular elements that. But in the finite element technique, what happens? You can of element. So that is the meaning of refinement of the mesh. So one triangles that increase from 382 to 1528, 1528. After that, again you can it is not required to generate more number of elements because you get the solution. The same solution you will get. Discretization of the domain, meshing of the domain, then refine taken from the literature. Here you can see the buffer association rate 1.6 to 600 micrometer per meter per second. Uh, is 0.1 micrometer. Why we have taken the range? Because if there is a different buffers are there, so define from where to where we can take. So what happens if in the first, if we talk about BAPTA is one kind of buffer is there. So if you generate the low and high buffers, so in the first phase, what means what? If the low buffer present inside the cell, then after the reaction, point, and then it goes down, 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 and it throughout the body, it goes to the zero point 0.1 micromole. That is steady state condition. It will take rest. Buffer consideration is taken considered higher amount, maybe the 200, 300 micromole. So in that case, what we can analyze from these two figures, if it is higher, yes, if the BAPTA amount is higher, so what will not be toxic for the cell. If we increase the amount again more, then what will happen? It will be in one cell to another cell. So we are talking about the pain. If the drugs comes in the chemical forms, medicine you are taking, response the neurotransmitter which is responsible for the pain, that is blocked. So billions of proteins also exist. So that's why we say the number of proteins are existing, and they are buffer is low, then it goes higher, higher, and in that case, what happens? The free calcium becomes toxic. If it is toxic, then what happens? Neuron partially dead is there, or some part of the neuron is dead, some part of brain number of neurons are decreasing. Forget to remember, many more things happens. Expression change accordingly. If we talk about the multiple fluxes, mathematically you can consider no issue. Biologically we have to prove. So after okay, so it comes from the different way. So in the different way, it is what it is higher, higher. If we talk about the higher amount, it will decrease very either the movement will not take place or very slow process will take place. Many more neurons and the movement of the neurotransmitter. Here we have considered the different buffers. Earlier it was so what is difference between these IGTA and BAPTA? It has the affinity is different. Affinity that we same way the multiple buffers we have considered here. So again you can see up to the high that way, and that it will decrease. Here also it at high and it is going towards about steady state condition. So if high amount of buffer is there, multiple buffer is there, it will block all the neurotransmitter. In this problem, I have tried to explain the role of which is a term in the protein that work as a buffering process that control the free such activities can be studied for the different kind of nerve cells are there. Uh, Cardiac myocyte is there, hepatocyte is there. Uh, Here, all these cells are working as a system. So in this result that we have applied the finite element technique, finite element is very good method for especially for the irregular geometries. If your geometry is regular, element technique, if we require to generate source and sink inside the cell, inside if you that we have taken earlier also, we have taken 9 months to solve this problem. If reticulum inside the cell and locations are also there, in that case also analytic method fails to give the answer. But location we can't add in the model. But once you get the solution, again you compare with the analytic solution because there is no question about the analytic solution. Neuron cell, complex mathematical formulation can be formed. Diverse parameter can be used to get a better insight about the disease with the help of calcium dynamics. Mathematically also on the fuzzy differential equation, uh, study of calcium buffering process that you can see also. And they have number of papers on this area. They have published number of papers in this area. For the mathematical term analysis, use the diffusion process.